episode 308 of the Stem Cell Podcast, Regeneration in the Cerebellum, Dr. Sumru Bayin. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Sumru Bayin from the University of Cambridge, Gurdon Institute. She's on the podcast to talk about her research on neural regeneration and just the complexity of the brain and development. We've also got our usual roundup of recent highlights in stem cell news that's coming right up.
But first, this week, we'd like to remind our listeners about Neural Cell News, one of Stem Cell's free bi-weekly scientific newsletters. Neural Cell News summarizes all of the latest research, news, jobs, and events in neural cell research and delivers it right to your inbox every other Wednesday. Save time and keep current with Neural Cell News. Subscribe for free at neuralcellnews.com. We've got a number of neural cell news relevant stories here in the roundup, except for one, which is something heart related, and I'll go ahead and start with that just to get out of the way. Not get out of the way, it's a great story, but it doesn't quite fit with the theme of everything else we're talking about here today. This is a story coming from Aitor Aguirre, who's really one of the leaders in cardiac biology over the years at Michigan State. First author here is Colin O'Hearn, and I believe actually a few years ago, last year or something, at the American Heart Association's Basic Cardiovascular Sciences meeting, which I like to go to every year, I believe Colin was actually presenting a poster on this. So it's cool to see this come to fruition. I know it's many years of work. The title here is Human Heart Macrophage Assembloids Mimic Immune Cardiac Interactions and Enable Arrhythmia Disease Modeling. And I think this has a lot of things that we love here on the show, whether it's in vitro disease modeling, whether it's assembloid and organoid technologies, it's looking at interactions between different tissue types. And this is something that's near and dear to my heart, pun intended, because it's something that we're trying to do in my lab, looking at immune cardiac interactions in these in vitro model systems, whether it's organ chips or organoid systems. And that's really what they're able to do. But they took it a step further and used their system to see how the immune system can influence cardiac biology and functionality. And basically at the tail end of this, they had a really nice arrhythmia model, right? So talking about the developmental side of things, these yolk sac-derived embryonic cardiac tissue resident macrophages, or TRMPs, these macrophages that are in the heart, early during development, they colonize the heart early in development in cardiogenesis, and you need them for proper development of the heart. The heart, if you have any knowledge of cardiac development, there's this intricate tube structure that forms that has a bunch of looping, and you think it's kind of a cardiomyocyte or muscle intrinsic process, but there's a lot of other cell types like neural crest and these resident tissue macrophages that also contribute to the proper formation of the heart during development. And these, auxiliary macrophages are important because they support tissue remodeling, angiogenesis, the electrical conduction system of the heart, of course, very important, and immune regulation, obviously, as well. So they aim to recapitulate that model in vitro and actually develop this human heart macrophage assembloid, or HHMI model, by integrating autologous human pluripotent stem cell-derived embryonic monocytes into the pluripotent stem cell-derived cardiac organoids. And they were able to generate this physiologically relevant TRMP that persists long-term and also contributes to cardiogenesis. So that's great. And they used single-cell transcriptomics, live imaging, proteomics to demonstrate that these TRMPs can modulate cardiac paracrine signaling, perform different functions, and regulate extracellular matrix remodeling, electrical conduction, so they're able to contribute properly to the different functions in the heart. And then they applied it. Like I said, the downstream application is really powerful here. They had a proof of concept matured HHMA model of chronic inflammation. And they're able, these TRMPs actually to adopt these pro-inflammatory phenotypes that can promote arrhythmogenesis, consistent with, atrial fibrillation, AFib, a really severe clinical situation here. So they're able to model AFib that was induced by these inflammatory phenotypes in this in vitro system. And then they took a deeper dive to show that it's not like receptor parent domain containing 3 or NLRP3, this one node that is regulating this inflammasome process. So it's a cool system because for one, you're establishing the system, you're establishing this immune cardiac co-culture system. And I know other folks, like for example, some folks from your neck of the woods in New York has also done similar work in this area, more on the immune cardiac co-culture in a chip-based system. But here's an assembloid-based approach to do that. And after you develop the system, you can look at these immune cardiac interactions and use it as an in vitro platform for studying not just human heart development, but also inflammation-driven arrhythmias too, which is a reality, unfortunately. So really like this study. It's, got a little bit of everything here, and selfishly it's something that we're trying to do in my lab as well. So I'm glad they got to it first.
Yeah, this is cool. They did everything, it seems. You look in the abstract, the highlights. I mean, it's each one of those could be a focal point in itself. I mean, with the exosomes, or EBs as they're called now, the paracrine element. Yeah, the imaging was pretty high tech. This idea of the assembloid of like distal, I know that like assembloids that we've talked about most of the time, well, a lot of the time, and we're going to talk about them in just a sec, are these neural assembloids which combine organoids of different brain regions, so same cell type. And I love these stories where the more distal cell types, in this case, the immune cardiac interface and relationship, which has a real important physiological, pathophysiological precedent. I think the thing with these, for me, the bar is always got to be greater than the sum of its parts, right? And I think they met and exceeded that in terms of the output here, in terms of what you're assembling and what the output is. The thing about these studies that to me gets more and more, I don't want to say confusing, but kind of overwhelming, is that, like, I like a simple model leading to a simple result that can be maybe generally applied or can be like a conceptual leap. Here, there's so many moving parts there's kind of like a dissonance or anachronism, I guess you call it, in terms of like you have these yolk sac hematopoietic derivatives, which sure, they clinically relevant for sure, and immature cardiac, and also missing a lot of the vasculature and other immune components, like which would be relevant in the context, I would argue, of atrial fibrillation, whatever the kind of pathology they're modeling here. So like as these moving parts get more complex and multiple, I guess while I do think they hit it here, I wouldn't know what to do in terms of where do you add to this model to get closer to physiology. I guess you start with trying to get mature cardiac cells or maybe more immune components, but I feel like the more you add to the wheelbarrow, the heavier it gets, and eventually the wheels come off, right, Arun? I don't know. It's tough.
That's a really good point. I mean, we're all so obsessed with making these models more complex, more, quote, physiologically relevant by incorporating different organoid types, cell types, and all that. But you're right. It just exponentially increases the amount of things that you have to dissect downstream, right? And it makes me kind of long for those really simple days 15, 20 years ago where we had isolated cardiomyocytes and we were just modeling something that was intrinsic to the cell itself, and you didn't have to introduce all these other cell types to look at the mechanism and look at the phenotype. I still think there's a power and a utility to have those mono cell type systems, even independent of an organoid system, whether it's just two-dimensional cardiomyocytes in a dish. Because, yeah, if it's a single gene causing an arrhythmia, causing AFib, then you can dissect that using a single cell type. And maybe you don't have to go ultra complex and build all these different, complicated assembloid types. So, it's kind of the catch 22 of the modern era of stem cell biology is, yeah, we want to make things more physiologically relevant, but you can't forget where you came from.
Absolutely. And very relevant now in terms of these non-animal models emphasis. I think, I don't want to say the cart's ahead of the horse. Maybe that's aggressive, but I feel like we need to, detach. Yeah, retain an animal model, develop the in vivo or in vitro models, but I don't think either of them is going to stand alone. But amazing study, no shade, really ton of work, and that's kind of what you got to do to get into cell stem cell nowadays, right? Is you got to have that clinical relevance. Staying in cell stem cell, I got a story that more assembloids here. I mean, apropos, it's kind of what you got to do. Can't come with those simple single cell intrinsic. Poo poo. Arun, what are you even talking about? Dinosaur. Anyway, this story is in the neural space, as I kind of alluded to, as Arun alluded to. We're staying neural for the rest of the roundup here. Very excited to have such good alignment with our guest. Probably going to delve deeper into some of the things we introduce here as we talk with our guests in just a moment. But first, yeah, here talking about complexity. There's so many different cell types in the brain, and I think one of the things that's amazing about these assembloids is that, as I alluded to, there's some of the parts idea is that you can get these unique cell types by combining two more simple cell types that maybe standalone you wouldn't get. In this case, what we're talking about is the nucleus basalis, right, which also known as the nucleus basalis of Meinert. And like that's, I say that because there's all these like subcell types, this Meinert, this guy, this girl, everybody's got a cell type in the brain, it seems, except me. But the point being is that there's a lot of cell types to go around. And this one, this nucleus basalis, is one of the primary cholinergic outputs of the basal forebrain, which is known to dynamically modulate cortical activity and plays a critical role in learning and memory processes. Dysfunction amongst those processes can be implicated in like cognitive impairment in the case of like as much as like neurodegenerative, like Alzheimer's, but also genetic disease like Down syndrome, cognitive, writ large cognitive impairment can be driven by dysfunction, this cholinergic pathway from that basal forebrain to the cortex, right? And of course, this is opaque. It's hard to get into these regions, particularly in human models, and to study these cholinergic neurons from a disease standpoint, understanding disease mechanisms, but also therapeutically, right? If you can generate cells that you can put back in, enter stem cells and organoids. So there have been a lot of region-specific brain organoids. We've talked about a lot of those, and also assembloids, right? Which allow you to model the crosstalk and synergy and circuitry. And also separately, you can transplant these brain organoids, right? And that can allow you to get these kind of extended functional assays in vivo. So there's a lot you can do with these. And in this story from the group of Yan Lu at Nanjing Medical University in China, they developed this approach for differentiating these pluripotent stem cells, human, into these neural nucleus basalis organoids. And I mean, another story with tremendous complexity. They actually ultimately put these, they generate protocols, but also did co-culture with fetal brains, human fetal brains to show like long-term connectivity. They ultimately settled on this entirely in vitro assembloid model where they combined the nucleus basalis organoids with cortical organoids. In vitro showed that there was a crosstalk. also transplanted those into mice, and showed that when you generated these assembloids, they could get these cholinergic neuron projections into the cortical organoid. So from the nucleus basalis organoid to the cortical organoid and form a synaptic connection. And then going even next level with the disease relevance, as I alluded to the Down syndrome earlier, they took these Down syndrome trisomy um, IPS cells and showed that those cholinergic projections were deficient in with that background as a model. So showing the modeling, showing like the cell type in the first place, showing that you can get this greater than the sum of its parts with these assembloids, and recapitulating the in vivo complexity of brain development. We're not just talking about the layers, but all the different cell types that have to be coordinated and to establish the circuitry underlying all the different brain functions. In this case, highly relevant to cognitive impairment, which is something that's been on my mind lately, or I should say, slipping my mind lately. Arun, what do you think about this story?
On your mind, I see what you did there. You think you're so smart. Yeah, this is great. Another example of the power of assembloids, just kind of building on the paper that I talked about at the lead off there. And I mean, they do say it. There's incredible advancements here, especially when it comes to modeling, potentially cognitive function and brain development, all this good stuff. I mean, a lot of advances, but still things that we have to improve. And ultimately, like we were just talking about, if the goal is to perfectly recapitulate that in vivo architecture, perhaps I'm pessimistic in that we can ever get there. I really don't know, but there's still things you could do, even in vitro, to push towards that dream, right? Which is impacting the full recapitulation of these CHNs, both their functionality, their connectivity. You can enhance the organoid cultures further, such as something like doing like what we always talk about, incorporating vascularization potentially to help make these organoids grow, take them to the next level, provide adequate nutrient sources, all that good stuff. That's going to help better approximate that physiologically relevant in vivo environment, right? Promote higher maturation. So you need that for the future studies. And one thing they also didn't do here, and I think this is relevant to some of the work that Sergio Pasco is doing, in terms of connecting these different brain assembloids and stimulating them in different contexts. They didn't address signaling inputs to their MBM model that they had here. So I mean, that's again could be addressed through a incorporation of another organoid type, perhaps, right? Like I think back to that paper that Sergio put out about the skeletal muscle organoid combining with the brain organoid, and how you can use that as like an integrated circuit, an all-in-one circuit for looking at input output electrophysiological responses. So I don't know if there's more stuff that can be built into this particular model, but perhaps they're looking into it already.
Yes, I mean, I can recall this was back in Hamburg, so more than a year ago, but talking to Sergio Pasca about just the state of the art. And he was saying that was the frontier now, is the circuitry. And everyone's showing the cell types and even the projections. But you really got to show that electrophysiological input output, as you just said. But I think what's great about this model, and it's clear why. I don't know if it was to address a reviewer, but I can see the rationale for including the fetal experiments here, because neural cells uniquely, maybe hematopoietic cells would be the other model, which is kind of the gold standard in terms of stem cell transplantation, right? But neural cells have this capacity for projection, and the fetal results here show that the neural projections of these cholinergic neurons were extensive over significant distances at that scale. So I think that while you're right, there is, I think, a higher bar perhaps in terms of showing that circuitry. The rationale for neural cell therapy, I see why neural gets most of the funding for the NIH, at least when it's open, because neural maybe has a greater feasibility in terms of transplantation, not to mention the unmet need there. But maybe counterintuitively, neural cell transplantation, because of that capacity for axonal projection, for some diseases at least and conditions, may be the move.
Yeah, we'll have to get Stefan Arian and the Blue Rock folks back on here on the show to see what they think about that comment. I don't think anybody thinks it's particularly easier than, other cell types, but I see what you're getting at there. Moving on to another brain-oriented story. This is an atlas, and atlases, as you know, are all the rage these days. But it's more than just an atlas. It's a brain organoid atlas. It's incorporating a couple of our favorite things here, and this is also importantly um, work that's funded by CIRM, California Institute for Regenerative Medicine, is actually using a number of the IPS lines that are in-house at the CIRM repository. So I don't know what the current status actually is of that CIRM IPS repository. It was pretty substantial. I mean, I think a few hundred, if not 1,000 IPS lines, but I think they're trying to find a new home for it. Maybe we have to have somebody from CIRM back here on the show to tell us more about that. First author here, Lu Wang. Last author here is Joseph Gleason from the Department of Neurosciences at UCSD in California, of course, right? So for the goal here, and it's important to look at the big picture here. The goal is to look at the genetic drivers of neurodevelopmental disorders or NDDs, right? And there's thought to be thousands of different genes associated with these various types of neurodevelopmental disorders, but mechanisms are elusive, treatments are elusive, and to fill some of these gaps, this is exactly what they did. They created a CIRM-driven, CIRM-initiated neurodevelopmental disorder biobank of, from here, 352 different publicly available, genetically diverse, importantly, patient-derived iPSCs, right? 350 plus lines, and associated with those lines are all the appropriate clinical details of that individual, the brain imaging, genomic data, and these individuals represented four major categories of neurodevelopmental disease. They had microcephaly, they had polymicrogyria, epilepsy, and intellectual disability. Pretty broad category there, and I think that's part of the issue with modeling some of these neurodevelopmental disorders, and I think Sergi would agree, is that sometimes the categories of classification can be somewhat ambiguous, like intellectual disability. That's a pretty broad swath right there, right? But regardless, that in itself was powerful to generate this NDD biobank. And then from there, they took that, took a little bit of a narrower approach and picked 35 different representative patients, which is still a very heavy lift, 35 different IPS lines, where they created brain organoids from them, more than 6,000 of them, and then examined them for their histology, their single cell transcriptomics, all the usual stuff that you would want to do. And then they compared those 6,000 brain organoids to an organoid library from what they called, quote, neurotypical individuals, which, again, it's sort of an ambiguous statement, right? That phrase, neurotypical. And when they did that comparison, the patients showed distinct cellular defects that were linked to underlying clinical disease categories. The microcephaly, which perhaps is the most extreme of those NDD phenotypes, showed defects in cell survival and excessive TTR positive cells. The polymicrogyria organoids showed intermediate progenitor cell junction defects. The epilepsy organoids showed excessive astrogliogenesis. And then the broad intellectual disability organoids show an excessive generation of those TTR positive cells again. So it's interesting because you have unique phenotypes associated with the organoids that are derived from each of those classes of individuals. So perhaps there's like a disease classification sort of thing going on there that you can uncover by creating these organoids. And of course, there's also extreme utility in the IPS lines that were generated for this study in the first place. So the atlas is demonstrating both the conserved and the divergent neurodevelopmental disorder category specific phenotypes, just like I said. And their goal, and I think they did that to some extent here, was bridging genotype and phenotype in these organoid models. So ultimately, just like is the objective of CIRM itself, they want this to be a resource for not just for the California community, but for those of us around the world who are interested in studying neurodevelopmental disorders using IPS models. And I've said many times here on the show, I think developmental modeling is perhaps the best application for iPSC and iPSC derived organoids because you don't have to necessarily worry about the maturation. You're recapitulating the developmental trajectories in a dish, somewhat analogous to what's happening in vivo. So hopefully this can support future developmental studies, maybe future disease modeling studies, and maybe even some therapeutic approaches as well.
Yeah, hats off to this group because what a heavy lift, and it's a kind of act of faith, I would say. I mean, in some ways, no, because you know that no one else, like, I don't think you're looking over your shoulder when you're doing this study, being like, oh, who's going to get this? Is there another group out there that's looking at 352 publicly available, highlighting 35 and doing 6,000 brain organoids for each with single cell? Like, they certainly um, committed the resources here to creating a vast and deep resource, which you assume um, will serve as kind of a fertile ground, a reservoir of hypothesis generation. But yeah, I just have to I'm kind of like awed by the commitment and the moving parts. I was looking into the methods here just quickly, just to be like, how did they automate? Like, how did they handle this without an author list that was much greater? But it's just this is what we do, I guess. A bunch of sleepless nights and long experiments. Although, as you said, we're not talking about like maturation beyond a year, which some people are doing with this neural organoid space. Still, I mean, it's a ton of work. So, hats off, and I'm sure a lot of people will be tapping this resource and moving forward.
Yeah, for sure. I think to make this a feasible study, you have to make feasible timelines, right? You can't have the Paula Arlotta approach of growing these things for years and years and years because the study wouldn't end, right? At that rate. Of course, there's a utility for that. It's a different scientific question, but another utility for this particular study. And also just a reflection on the power of CERM, right? They've been doing and leading these pioneering, large throughput studies for 2 decades now. And CERM was founded on the promise of basic science. And this is, I think, first and foremost, a great basic science study with, some applications for studying human disease, right? But I will say that, and hopefully this isn't controversial, the most recent iteration of CERM has certainly taken much more of a clinical angle. And I think
Mm.
even so many more of the grants that are available these days are clinically and translationally and clinical trial oriented. And I get it. Um, Part of what CERM needs to do these days is to ensure its survival. And to do that, they have to show that the work they're funding is leading to new therapies because that's what the public, the California public, which ultimately funds CERM, wants to see. Of course, everybody loves the basic science, but people want to see treatments and cures. And I think that's if CERM is able to survive into the future, that's fortunately or unfortunately the angle they have to take.
No, for sure. And no coincidence why microcephaly was among the more closely analyzed phenotypes, because it was so stark. I mean, you look at the pictures and clearly there's something going on there in terms of the organoid growth. So yeah, you really want to capture the power of these systems. But I'm glad you highlighted the importance of these big institutional efforts by like funding bodies like CERM, also like Allen Institute and others. They really keep the machine moving. Finally, we got a kind of a different kind of atlas in terms of, very focused and open-ended and a resource in its own right, but very focused on a different kind of analysis, right? And this isn't the first of its type, but I would say that predominantly we talk about these atlases, and what we're talking about is snapshots for the most part from whatever, I don't know, primary cells or tissue or organoids. But the idea of the lineage tracing within that, it's you kind of take a bunch of snapshots sometimes, and then you put it all together with monocle or whatever kind of trajectory analysis. We just talked about one of those last episode. But doing bona fide lineage tracing involves a lot more tech. Oftentimes, you need to have these genetic models, model animals, right? And we do have those in mice in particular, and from mice in the brain, at least, these clonal lineage tracing studies have increased our understanding spectacularly, right? For example, because of lineage tracing, we know that radial glia in the ventricular zone in development act as like neural stem cells, right? And they differentiate according to this hierarchy that's not only based on the layers, deep or upper cortical layers, but also based on the array of cell types that they generate: astrocytes, oligos, even these olfactory bulb GABAergic neurons, which we don't really talk about a lot. But in addition to the astrocytes and the oligos, you get that kind of cellular purview, right? But extending these studies to humans, even to primates, it's tough, right? Because a few reasons. One, neurogenesis in primates and humans is much more protracted, takes a much longer time, obvious. And related to that, the cortical progenitor cells in humans, they have a disproportionately large numbers of these GABAergic neurons that I just referred to. And also, similarly, because of that protracted development in the human cortex, there's a distinct subtype of these radial glia that emerge much later during the second trimester of development, which, much later, not only in time, but also in terms of like maturation of the brain, like the human primate brain relative to the mouse, a different animal, pun intended. So here we have this novel, massively parallel lineage tracing approach, they call it. This is coming out of the lab of Tomas Nowakowski, who's at UCSF in San Francisco. And it's such a cool approach. Involves like this lentiviral labeling. It's like a error correctable, so that you can. I mean, it's a lot of tech that I haven't quite looked into to understand, but it's dope. The cooler thing in this study is the technical approach here, which I just want to talk about for a hot tick, because they took human brain, first of all, human fetal brain, around that time, second trimester, where you get the radial glia. And they did this slice culture. So first they did slice culture, but then they dissected out the ventricular zone and the outer ventricular zone, took it out for a sec, took it away and hit it with this stick. It's called sticker is the acronym that they do this massively parallel lineage tracing. And then they put it back into that slice and culture it for like 10 to 20 days. So an idea there is that they want to label these things, they do single cell seq on it, right? With the lenti, and then they're able to reconstruct these lineage trees as these cells migrate from that ventricular zone and go to the outer layers. So it's a really clever way of doing this kind of ex vivo, in vivo, I would argue highly physiological lineage tracing in human tissue, like at scale, right? So here I think they had 9 brains that they fetal brains that they took, 4 or 5 that were early, and then 5 or 4 that were later stage, and they did this lineage map that linked the progenitor cells to the specific subtypes of neurons and glia that's like been the black box that we couldn't really exactly reconstruct the relationships there. You could get the snapshots, as I said, but you couldn't specifically trace those in humans, even primates. We're not there yet where we're doing that kind of genetic lineage tracing. Anyway, they profiled the clonal output of around 6,400 cells. Um, and here's the key, like, this wasn't just like, hey, look at the data, this is what happened. They actually gleaned some really important insight. They showed that the cortical progenitors switched from this glutamatergic to GABAergic neurogenesis. The timing of that is around mid-gestation, which coincides with the timing of the oligo generation. And they find also, and this was cool to me more conceptually, it relates to, I think, all of our systems, is that these truncated radial glia, they retain this glutamatergic neurogenic potential for a long while, right? They hang on to it into the later stages of human cortical development. And when you spin off these glutamatergic neurons at a later stage, these late-born glutamatergic neurons, as they call them, from the radial glia, they're like of a different type. And the functional relevance of that is that they may be critical to the expansion of the subplate region during mid-gestation. So, like, that's a lot of neural stuff that, frankly, I'm reading. I don't know a lot of that in terms of anatomy, cellular, physiology, molecular biology. But for me, what's so cool about that is this duality that, like, development is mushy. The anatomy, the fidelity, you get the same thing over and over, but that's because you have this kind of mushiness and this outside the lines kind of, we all in our mind picture these clean lineage dendrograms, but the reality is, just like in the hematopoietic system where it's not so clean, there's a much more of a I would say, plasticity along the temporal dimension for these cells in terms of what their purview is. So I love that idea because I think it opens up my own models where I can start thinking of it as like, okay, I have windows within which the epigenetic profiles of these cells enables their differentiation to whatever subtypes. And those windows are maybe not so circumscribed as we think of them in our classically drawn lineage trees.
Development is mushy. I love that. No, I think it's true, and it is a reflection on topics that we've talked about recently here on the show, how developmental trajectories are perhaps not as concrete or set in stone as we traditionally read about in our textbooks, right? There is mushiness. There is intra convertibility from cell progenitor to cell progenitor that happens in vivo, and sometimes you can approximate that in vitro. And I think the newer textbooks are hopefully gonna reflect that. I really like this study, obviously from the technology point of view, but also from the evolutionary development point of view, right? Understanding the species specific differences in neural development. I think that's one thing that they're able to get at really well. And in particular, like for example, their data point is showing that there's a continued production of glutamatergic neurons that are expressing these subplate neuron markers in certain mammals that, that is again species specific. Whereas in rodents, these subplate neurons are among the first neurons to be generated in the cerebral cortex, but in primates and humans, this process could take place throughout the corticogenesis process. So there's timelines here that are different between different species, and I think this study helps to kind of get to the bottom of that. And yeah, perhaps this second trimester is so critically important for developmental transitions, right, in cortical neurogenesis. And maybe this is a part of the reason that we're humans and mice are mice, is because of some of these alterations and differences in developmental trajectories between the species. So I really like it from that angle too.
Yeah, and I'm glad you underscore that. As parents, we appreciate that, brain development extends way beyond even pregnancy and childbirth. But, I think the focus has long been on anatomical endpoints, neural tube closure, et cetera, where, I just think, although we all knew it already, just all the foundation of our thinking brains is really set up along a pretty extensive trajectory. involving a lot of moving parts. It's a lot to wrap your head around, man. Neural is crazy. We're going to talk about that with our guest Sumru Bayin in just a minute. But just before we get there, I have a quick message from STEMCELL Technologies, who's inviting you to find a pathway into the peripheral nervous system with STEMDiff Neural Crest Differentiation Kit. Whether you need Schwann cells, sensory neurons, or sympathetic neurons, STEMDiff Neural Crest Differentiation Kit enables high purity generation of neural crest precursors from human pluripotent stem cells. Learn more at www.stemcell.com/NCKIT. All right, everybody, joining us for this episode, Dr. Sumru Bayin, who is group leader at the Gurdon Institute in the University of Cambridge. The Bayin Lab studies the mouse cerebellum as a paradigm to identify cell intrinsic and extrinsic mechanisms regulating key stem cell behaviors during development and molecular factors that inhibit effective regeneration in the adult brain or support regeneration in newborns. The ultimate goal in the lab is to discover how the regenerative potential of the endogenous neural stem cells can be harnessed to address human disease. Sumru, thank you so much for joining us on the show today.
Thank you.
Yeah, so Dalen kind of gave an overview of what your lab is focusing on, when it comes to stem cell behaviors, regenerative potential change from neonate to adult, particularly in the cerebellum, like you alluded to. So we always like to give our guests a chance to kind of talk about their lab in their own words, give kind of their own overview of what they're doing. So give us that overview of what your group is working on, and in particular how you kind of see it fitting into these broader themes that we've commonly talked about here on the podcast lately when it comes to brain repair, aging, these super hot topics.
Yeah, no, absolutely. That was an excellent overview to begin with. But usually, where I stand in terms of how I think about my science is actually quite fundamental where we start, because I believe we're still lacking a lot of fundamental knowledge about what regulates stem cell behaviors, specifically neural stem cell behaviors molecularly, to be able to really utilize them for therapeutic purposes. And this knowledge is particularly limited outside of the known neurogenic niches where we have been studying neural stem cells for a long time. And therefore, we study the cerebellum as kind of our entry point. Not because of this, but also during my postdoctoral training with my colleagues had been showing, and then I started continuing on that work, that the neonatal cerebellum is actually quite interesting, and it is showing these lineage plasticity with injury type phenomenon that we don't normally observe elsewhere in the brain, or at least to date we haven't been able to pinpoint those behaviors, something we're in the future interested in the lab as well. So using the cerebellum, where we do see true lineage plasticity with injury without doing anything else, by studying this model, we're trying to understand how can successful regeneration occur in the mammalian brain, essentially, and then hoping that understanding can give us ideas about how to tweak when this is not happening so that we can generate new neurons. As you know, there are plenty of amazing research happening around for neuron regeneration from, I think primarily the most exciting ones are definitely the Parkinson's to me, where we are able to, transplant induced pluripotent stem cell or pluripotent stem cell derived neurons. But some regions of the brain, similar to cerebellum, where the structure is very complex or more than a single cell type is affected, I think not necessarily transplanting the correct cell type may not be sufficient for repair. That's why we're trying to understand, can we induce endogenous mechanisms to take over for repair, and can we make neurons, can we reactivate developmental pathways to make neurons and let them basically take over from that point of view. So that's kind of the beginning of the therapeutic thinking that we have, but we're still, I need to admit, we're really still in the fundamental understanding aspect of this. And we're really trying to understand, first of all, what are the key progenitor stem cell populations that can do these behaviors. We're still trying to understand what are their developmental relevance and how do they normally make cells. And also, what isn't specific about the injury and injury microenvironment and, cells that are dying that is triggering these injury-induced lineage progression, which is quite interesting, actually. So that's what the lab is focusing on at the moment, really trying to understand how does that particular neural progenitor know which cell to make, let that be in development or now with injury, and what transcription factors are specifically important for that decision making.
Yeah, I mean, when you think from an engineering perspective, unlocking the endogenous potential of the brain from a repair perspective just makes a lot of sense as far as we've come, with the exogenous cell therapies. It's very exciting, but yeah, why not tap that resource if you can? But more, as you just said, very eloquently, it really is about just lacking a fundamental understanding of that. You could argue the final frontier of cellular biology because of the accessibility issues. And we're going to come back to that and drill down on your science. But a big part of your science, any scientist's work, is your training, right? And as we were talking about briefly before the show, I can actually remember your name floating across my email for the many postdoc students stem cell fora talks that you gave while you were at MSK, Memorial Sloan Kettering, which is just down the street from me. And of course, you were in Alexandra Joyner's lab, who just retired. It was a big deal. Just retired this past year. She spent 40 years training pretty much a whole generation, multiple generations of innovators like yourself. And I think notably for me, as an outsider, the Joyner lab thrived. From my perspective, it seemed like they were really good at bridging that gap that can sometimes exist between like old and new school molecular cell developmental biology across the board. And so I will ask you, as a relatively new investigator who was trained in that legendary lab, what was unique about your experience in that phase that has maybe carried through to your own practice of mentorship and lab stewardship at Cambridge?
Yeah, no, that's actually a very nice synthesis of the Joyner lab, in the sense that gapping the bridge between very critical traditional developmental biology techniques, some of which have been developed within Alex's lab, all the mouse genetics, and bringing in new techniques, which I think kind of was what my initial projects involved, really implementing single cell genomics in this area where we kind of we hit the limiting point of what we can understand by just looking at the lineages or understanding the cellular behavior on histology or sections, what's happening to the progenitors and what do they generate, but how and what is the molecular parts, molecular mechanisms that drive this was kind of the limited technology at that point. So there we started implementing, for example, single cell genomics now back 6-7 years ago when we first started playing around with those ideas. So it's an excellent place to be, to kind of bridge very fundamental, very hardcore developmental biology techniques, fantastic, mouse genetics knowledge, and fantastic, particularly lineage tracing knowledge that Alex is kind of in my eyes, the god of that, essentially, to understand what how do different progenitors generate different systems. And my PhD was a bit more molecular, so there actually we were kind of a great team in the sense that really building up the molecular knowledge of these behaviors. So she was more interested in cellular and organismal effects of things, and I was more interested in the mechanistic aspects. But to be honest, as you can tell, some of these questions we're understanding is not like a simple, oh, we found the X gene and that's it. And no, we found the X gene and now we know it's even complicated because it has Y and Z as partners. So like a lot of it is still, even in my lab's work, we're still on the descriptive side to a certain extent and building on mechanism while we're describing cellular phenomena to most case. Alex was a fantastic mentor. I think she gives great freedom if you need to, but also she's always there to kind of guide you to the big questions. She's really good at identifying, the directions and the knowledge gaps, I guess, in the field. And her attention to detail is amazing. So I think those are the practices I try to maintain in lab, being very involved, not actually, being involved doesn't necessarily mean micromanaging. Like, you can be involved without micromanaging. And she was fantastic at that balance, at least in our relationship. So I'm trying to maintain that and, adjust, tailor my mentoring depending on, some people may need more input where others like the freedom, but, want to bounce off ideas. So I'm kind of oscillating between those extremes and different career stages, and with different mentees of different career stages.
Yeah, I mean, it's something as a new PI myself, I'm trying to figure out like what's that right balance of hands-on, hands-off. And I think the best PIs and the best mentors learn to be adaptable and try to provide their trainees with kind of a customized approach, depending on what they need at a moment in time. Different personalities have different requirements, of course, right? So, taking a little bit of a deeper dive into some of the work that you did in the lab and some of the work that you're doing, of course, in your own lab. I mean, you showed that in the neonatal cerebellum, so there are certain like nestin expressing progenitors that can reprogram, replenish, for example, granule cell populations after injury. But that, like what you're talking about, this regenerative potential is lost in adult stages. And like I draw the parallel as a cardiac biologist to the loss of regenerative potential in adult mammalian heart tissue, right? It's there neonatally, but then it's just lost over time thanks to a variety of different molecular mechanisms. So talk more about kind of the details of that process. Like, what are some of the key molecular epigenetic changes that might be able to restrain ultimately the plasticity in adulthood, if that's something that we want to get to? And how do you identify or reverse some of these phenotypes in your lab?
Yeah, no, that's a fantastic question. And we actually interact and collaborate quite a bit with cardiac regenerative biologists, exactly because of this phenomenon that we both see in both developing cerebellum and developing heart, neonatal heart. Yeah, so this is, I think, a multifaceted issue. So there is the cell intrinsic epigenetic silencing of, all sorts of stem cell proliferation differentiation programs. And actually, I'm hoping to submit a manuscript like any day. It's done. I just hadn't had the chance to proofread it yet. That compares our neonatal progenitors to their we've been able to characterize these rare but still nestin positive Bergman glia, which is the specialized astrocytes of the cerebellum. And these Bergman glia cells are basically in the neonates what our nestin expressing cells give rise to when there's no injury. But when there is injury, they're now able to reprogram and make new neurons. So although in the adult, a similar nestin positive Bergman glia, which is to some extent fulfills some neural stem cell characteristics. For example, when we pull them out, they can grow in spheres. They can form these clonal growth criteria, which people have been using. And they do not express some of the canonical mature astrocytes markers. So they do look a bit more immature compared to rest of the Bergman glial cells. But when there's injury, they can't regenerate. They increase in numbers, which we are still trying to understand the mechanism, but they cannot increase their proliferation and then all the, turn on fate switch programs and differentiate the neurons. So there is the, hypothesis number one is there are significant epigenetic silencing of these programs in the adult. And we've been able to demonstrate that. And in this manuscript, hopefully next week can be on by archive. We can see basically just even globally comparing the chromatin of the adult cells are dramatically, in closed chromatin conformation compared to the neonates. And this involves the proneural genes, the proliferation genes, anything that you would want a, happy, strong stem cell to do essentially. But I don't think that's the only side of the story. And that's something we're also starting to investigate more in the lab. Because when we think about how the brain microenvironment changes from the neonate to adulthood, it's also as dramatic as potentially sometimes we can argue even more dramatic than just the cell intrinsic changes. So at the neonatal stage, because cerebellum develops quite protracted, it's still a developing system. And actually, there isn't many mature astrocytes, which are normally in the adult brain, are not that great for injury because they would cause scarring, and scar is generally not regenerative. And alternatively, the other, the immune populations, the microglia, haven't fully migrated to the cerebellum, or there are, we think, and the people have documented there are dynamic changes in microglia populations over development and in adulthood. So we think they may be more pro-regenerative in the neonates, where it is more acting inflammatory and anti-regenerative in the adults as well. So while the progenitors are not able to upregulate the proliferation and differentiation or neurogenic programs on their own, they may also be actively getting inhibitory signals from the environment in the adult brain. And we think for a successful regenerative biology, regenerative medicine down the line to work, we need to tackle these simultaneously, and not necessarily via just a single magic molecule that will gonna give and it's gonna work. But we need to do a combinatorial and sequential approach. How is that going to work out? I haven't really figured it out yet. We're trying to identify the targets. But one of the exciting types of therapies, potential therapies, I must very strongly underline, is we're utilizing modified RNAs, because there's a reason we lost our regenerative potential. And we think that's partially if we constantly get aberrant reprogramming or, overactivation of these developmental pathways in adults, we would probably have tumors popping here and there. But so the molecules or pro-regenerative mechanisms we tend to identify could also likely be tumorigenic. I mean, this is, I think, general regenerative medicine field issue. So we're utilizing modified RNAs, which allows us to transiently upregulate genes of interest without really changing the genomic makeup of the cell. So we identified in a study in collaboration with actually neonatal heart regeneration biologist Kathy Wilson, also at University of Cambridge, that MYC and cyclin T1 is actually specifically, the combination is required for promoting proliferation in cardiomyocytes, but also now we know in our NEPs as well. So we are activating this via modified RNA, and we got our first initial in vivo experiments, and it looks quite promising. So we can transiently induce proliferation in our nestin expressing cells in the adult brain at the moment. So to be tested is that sufficient to induce reprogramming as well? Or then do we need to give another reprogramming factor like the proneural genes, which everybody has been working on to push it to the next step? So it's been quite exciting and rewarding to kind of tackling the same question in multiple different angles, while we're still trying to understand to what extent these are important in the neonatal case, and what is the clinical relevance also of the neonatal regeneration. Making this progress in the adult brain is quite exciting for us.
Wow. Juan Carlos at Altos Labs got his eye on you right there. Probably going to try and snag you from the Gurdon.
I should disclose though one thing in terms of brain regeneration. We don't really need to age that bad. We stop regenerating quite soon after development. So
Yeah,
we're actually for funding cost reasons, and also we don't want to spend years waiting for the animals to age. They're quite post 4 to 6 weeks, 8 weeks, 2-month-old max. We're doing this, so they're not really aged mice. But obviously, as we delay the process, it only gets worse. So we need to just make sure we can hardwire these cells in a good way.
The principle there, I think, is what's so as you said, rewarding, exciting. And that is the core principle of your work, is this differential potential between the neonatal and adult state. And as you led in the jump with, you're still working out the basics and the mechanisms, just trying to understand how the system works. But there, I think, as you just described, it really underscores the power, clinically speaking, therapeutically, of how you can tech it up a little bit to hack those mechanisms. And there's this neonatal adult duality that you're describing, but also you have this other paper that's already on the bioarchive right now that has a different sort of duality at the center. But similar idea, right, as you just kind of alluded to there, is balance, right? You can't have these things firing throughout adulthood because there's going to be a cost, of course, metabolically, but also just in terms of the liability, tumorigenic potential there, right? In this posted bioarchive paper, you talk about the differentiation of inhibitory neurons in the developing mouse and also human cerebellum, whereby proliferation and then survival/differentiation is governed by ASCL1, which has been an old friend of yours, and FOXO1, respectively. Elaborate on this, and maybe more than the details of the study, because it's posted, is like what it means, what that kind of means in terms of conceptual, also maybe clinical, if you were to hack it, but also just, the conceptual underpinnings there that I think are fascinating.
Thank you. That's very exciting to hear. So that's our lab's first independent paper, by the way. So I'm
Congrats.
quite proud of it.
It's a good one.
Thanks. Yeah. So, and this is what I was kind of trying to bring it up earlier, in the sense that, we think we identified a factor, and then we just, in the next study, we know that's alone, its regulation is not sufficient enough to understand that particular progenitor behavior. And so one of the challenges generally is to understand really the gene regulatory networks that drives the lineage decisions. And yes, we know, for example, ASCL1 has been, even before our study on the neonatal regeneration, it's fundamental for inhibitory neuron generation in the cerebellum. And then in our study in the with the injury, we show that it is actually one of the required transitory states for the glial progenitors to now make neurons with injury, and it is an injury-induced cell state. So one of the questions that's been bugging me, and I still don't have the answer about this bioarchive paper, is one thing that we are trying to work towards. It is that how is the same transcription factor in two different contexts leading to very different outcomes? So normally it's making developmental inhibitory neurons. In the other context, it's also now, although pushing cells to an neuronal fate, it's making a neuron type that normally developmentally is never involved in this excitatory granule cell progenitors. So there is obviously the what type of genes what target genes it binds. It's one of the questions, but what regulates what it binds? We're just talking about a fundamental basic helix loop helix transcription factor. So there we started playing with the idea, and this has been documented in different contexts. And it's not just like the idea is not as unique, maybe, but that there can be additional transcription factors that regulates binding targets and also kicks it off, so that transcription differentiation paradigms can follow up. So to understand that, we started looking into single cell and trying to build these in silico gene regulatory networks to understand from the progenitor state to a more differentiated committed cell type, what are the different transcription factors involved. And there we came across FOXO1 and the inhibitory neurons. And this is important in the developmental context, because now we know through our study, although some of it needs to be validated in vivo, it is required for inhibitory neuron production, which actually regulating the numbers of correct neuron, different types neurons, is very important for building the right cerebellum. And therefore, the interplay between ASCL1 and FOXO1 turns out to be critical to the output, the number of neurons that can be either made, and they regulate different processes to ensure sufficient cells are made. But what we're thinking about this is potentially absence of FOXO1 in the glial cells is what is able to make ASCL1, injury-induced ASCL1 now to give out a different fate. And we're trying to now play around with overexpression experiments with or without injury to see if this is correct in the other progenitors. That's something we haven't been able to do. But the other exciting conceptual thing about that particular bioarchive that I'm very interested in is, as you pointed out earlier, all our understanding is in mouse. We're talking about mouse, neonatal mouse brain. We're talking about neonatal adult mouse brain and how these are changing. And we still don't know whether the human cerebellum can regenerate, or is that a critical neonatal developmental perinatal window that human cerebellum does show these behaviors. And obviously this is not something straightforward to study, but in the UK, we're fortunate enough to be able to get fetal cerebellar tissue, and we can actually generate the same primary lines. And part of the paper is actually thoroughly characterizing these primary lines. And we can then do some in vitro experiments and at least test whether the gene regulatory networks we identify in mouse are conserved or not. And in parallel, we're building some organoid models to see if we can see, at least in vitro, some of the lineage plasticity we're observing. That's exciting work, but there's a lot more that needs to be done on that front. That was what I spoke at the ISSCR last year. Yeah, so there's a lot, the unknowns multiply logarithmically when we try to translate some of the neonatal findings to human, partially because also the accessibility and the materials.
Yeah, I mean, a lot of stuff that's going to keep you guys busy for years to come. That's for sure, and especially as these technologies continue to evolve, it's going to be fun to play in your lab, I suppose, right? Shifting gears a little bit, you also put together this little commentary that was really nice. It was recapping a recent workshop that I believe you led or contributed to. It was published in Development, I believe. Building and Rebranding Complex Tissues Strategic Visions from this research-led workshop. That was the name of the commentary. And the focus, I think, was, figuring out how you can integrate these diverse regenerative contexts like we're talking about from both non-model organisms, model organisms, and extending some of these findings to potentially even organoid models, right? Where you can use them to study mechanisms of regeneration. And, certainly we can do this in a cardiac context as well. So tell us more about this meeting and kind of what you guys learned from it, especially when you took this comparative lens, right? How do you take these lessons from these highly regenerative tissues or even species to guide your thinking about neuroregeneration in the brain? And how do you extend some of these findings to like the model systems that we talk about so much here on the show, like organoids, for example?
Yeah, no, that's been a quite rewarding interaction that we've been doing under this complex tissue regeneration group. And so this commentary was based after UK-wide, but we had a few international attendees as well, meeting that we had where we invited regenerative biologists who work on very different model organisms, or all sorts of animal spectrum. And the first day was a traditional symposium, 10-15 minute talks, poster session, the usual. But then the second day we had the closed door workshop with the invited speakers and also the regenerative biologist community we have within the University of Cambridge. And the goal was to come up with open questions in the field. And we kind of in that commentary identified 4 main areas of open research, and also to identify the challenges and roadblocks on what's preventing us, either by technology or whether the lack of funding mechanisms that is able to fund these type of descriptive, cross-species comparative type studies. So we had a range of, developmental biologists, regenerative biologists, computational and modeling folks who were all involved in this. And we all have our favorite regenerative and non-regenerative phenomenon. So basically, what are the fundamental principles of what makes you regenerative, right? What is there global formula and what is, universally we could expect that's a regenerative, check mark, or whether there are species specific. Obviously, there will be both of these, species specific or tissue specific, across these different scales. What are the unique and overlapping characteristics is the main question. And how do we unify isolated knowledge that's generated across different labs in regenerative biology to understand this holistic, mechanistic, understand the background of that. And, this is important, and this is still on the fundamental side of things. And while we build up that knowledge, then we can identify key, for example, like pro-proliferative controlled ways to induce proliferation as a potential therapeutic approach, or like a potential modeling that we can predict regenerative outcome from other mechanisms or other tissue types that could be useful. So these are kind of the broad ideas that was bounced on and off, but it all comes down to funding. Basically, essentially, this was discussed on how can we, lobby for funding, or it all comes down to people, to unite people to get, new students or postdocs interested about these questions to take on these type of roles, and try to get them also funding or studentships. So that's kind of the other part of the discussion. So what types of packages that we can put together that could be attractive for funders. So it was quite fun exercise. Also, we did like scientific speed dating. We did like walks around. So it was quite fun, and it was organized by my two close colleagues here, Ben Stevenson and Michaela Stroher. So I just need to give them credit.
So fun. Fun, and it comes down to funding. I mean, no surprise there, but the way you guys collaborate on that, I think is shows the kind of more fun, as you said, fun side of science. Talking about this show, I mean, we're pretty agnostic, I would say, when it comes if it's stem cell related at all, we get it in, right? But I have to admit, we have a bit more of a focus on pluripotent stem cells, if I'm honest. But as you were just describing there's so much in these systems in the adult that are really fundamental and also complementary to these pluripotent stem cell systems. So, no surprise that there's renewed enthusiasm in that adult stem cell space. In fact, I would say, ironically, we're talking more and more about adult stem cells over the last few years. And of course, notwithstanding the amazing progress that we're making with these exciting trials of pluripotent stem cell derived products in the neural space. But as you kind of illustrated really well, approaches that leverage the endogenous potential of these adult stem cells, both in the brain and elsewhere, are gaining momentum, I would argue. And that's a real long way from when I got into science. The dogma was still that there was no such thing as adult neurogenesis, much less cardiac neurogenesis. So you young scientists out there don't know what kind of dark ages we were living in our ignorance. And you've shed light on some of that darkness, Sumru. But you talked a bit about how you might deploy this modRNA approach a bit earlier. Is there a comp? Like, are there existing approaches in the field right now that tap in adult stem cells, like a reactivation approach or other? If maybe you could talk about those by way of like, what are the major challenges that face widespread or efficient practical deployment of these types of therapies?
Yeah, no, absolutely. Just before I answer that, I want to say, I would have loved to have a very reliable organoid stem cell model, but our challenges, they're still so immature. And this is something we're actively working in the lab. And it is a different biology almost we're investigating when we talk about cerebellum. The stages, although, we still can come up with reasonable hypotheses and conclusions, it's really very embryonic fetal stages we're talking about, not anywhere near what we're talking, but we're still working on them. I think it needs to be, in certain fields, a good complement of both. In terms of modRNAs or activation of approaches, so I mean, we've been all part of this modified RNA experience with COVID vaccines, essentially. That's exactly what we've been given, and with the jabs, and it has been in clinic and vaccine context. In terms of regenerative context, as far as I know, it hasn't been applied to a degenerative or injury-based system. I know some companies are working on it, like in the cardiac field. I think it's much more advanced than what we've been doing. Basically, this was something we came across through actually one of these complex tissue regeneration group meetings with Kathy, where she was using on cardiac injury model. And we thought that would be quite interesting to see in the brain as well. So I think there's a lot of new momentum on that. And I know a lot of pharmaceutical companies are interested in RNA-based therapeutics, essentially. But there are challenges, and challenges are big. First of all, the delivery challenge, right? So these are, we have been lucky in the sense that it seems to be with our classical lab-based, lipofectamine type of injection transduction methods, it does specifically affect our Bergman glia cells, which is the only cells we want to reprogram, but that was pure luck. And obviously injecting a lipid-based thing that breaks membranes in the brain may have some challenges for what's healthy remaining. And the other challenge is actually, while we're trying to make them transient, sometimes it can be too transient, right? And then you don't want to go in and multiple injections into the same site over and over to give the modified RNA. So there are variants that people I know have been working on to increase the stability of it, and increase the output or the protein output from that. And that's a bit like the temporal dynamics needs to be regulated and may not work for every regenerative phenomenon when the regeneration needs to be quite extended period. So those are some of the challenges we need to think about. And as long as I think activating these pro-regenerative, pro-proliferative, pro-differentiation genes, as long as it's done in a controlled way, that may be through modified RNAs, but also like potentially just small molecules as well, as long as it's controlled and we know it's cleared well, I don't think it poses too much risk. Because usually for example, one of the other molecules we're interested in the cerebellum context is sonic hedgehog, because in the neonates we know hedgehog activation is crucial for both these progenitors to proliferate, but also the regeneration as well is one of the other mitogens. And in the adult, but on the downside, in the neonates, it's one of the key drivers of medulloblastoma overactivation of the pathway, the brain tumor, the pediatric brain tumor of the cerebellum. But essentially, if you activate hedgehog in the adult brain, there isn't any of those progenitors, adult cerebellum, there isn't any of those progenitors left to become tumorigenic. So not necessarily it would immediately, unless there's some second hit of some sorts or some weird transformation, it shouldn't necessarily with one of or two of treatment lead to tumorigenesis, in my opinion. So we need to really understand that control essentially, and it again comes down to really understanding which type we're triggering in vivo, right? Like have the opposite, like we need to know what we're putting in exactly and how well controlled and so on. If we try to activate some cell type, like we want to have control of what is that cell is and what is the activated component, or what is the after that process that cell is, what is that happening to it over time, is quite important. And then the biggest challenge, in my opinion, I think these are more attainable in my vision, but once we make that neuron, okay, I think hopefully we can make the right neuron type and abundance, let's assume that. So how does it go back to the circuitry, and then while this is happening, how do we secure the rest of the circuitry, because these cells all rely on each other to survive as well. So I think that is still in brain structures where there's a lot of cytoarchitecture and a lot of structure to how the circuitry is built, and like temporal order of how the circuitry is built, I think that's going to be the next big challenge, because it's not just a nuclear structure where you can throw in a bowl of cells and they will at some point connect to something. It may that may not be the case in the cerebellum, where some of the cell types needs to be at the correct layer and communicate with the correct subset of neurons, as opposed to the others may need to do. So that's my general big challenge. And I think this is I'm usually surprised why people don't push me more on this, to be honest.
I mean, you're at a place where you're definitely going to get pushed on this. I mean, you're at an incredible institute when it comes to the Gurdon Institute and the Cambridge Stem Cell Institute. Of course, bit of a bittersweet moment these days with the passing of Dr. John Gurdon, of course, who's an icon in the field, and he's definitely very much missed. But you're at a great place to do this kind of work. You're at Cambridge, right? This amazing ecosystem of developmental biology, such amazing history there, and stem cell science, neuroscience. So what is it about being at the Gurdon Institute and the Cambridge Stem Cell Institute that you find especially exciting or enabling for your research? And in addition to the history of the place, of course, but what's it about the environment and the people there that's just an awesome place for doing science?
Yeah, I mean, I think there's a lot of appreciation for basic biology and, trying to learn the fundamentals in Gurdon Institute. And very much makes me feel both intellectually challenged and also, in makes me try to want to understand and solve the fundamentals of how these making processes are made, and so that we can apply this in a therapeutic way. The best thing about Gurdon Institute is actually none of us really work on the same phenomenon. We all work on different organ systems or different organisms, all sorts of different scales of it. But I think the overall trying to understand how development and disease mechanisms are overlapping, and by studying development, how can we understand disease mechanisms? And now with our future new direction towards understanding this in a human context is kind of where I fit perfectly in my own sciences vision, where we're trying to use this mouse cerebellum as an entry point, but how do we translate this to human health and human disease? And there I feel very supported, because my neighboring lab is interested in the similar question, although it's in a lung setup or cardiac setup or all sorts of these different things. So there's a lot of intellectual exchange, and we learn from each other all the time. Obviously, there's an emotional connection. John Gurdon's experiment was my favorite experiment, so I was very excited when I got an interview offer to begin with, and then now being there. And he's a very inspirational figure. He still came to lab until the quite very late before his retirement till last year, and just having fun with science is having fun with questions, and just trying it is quite I can see it as a style in his approach towards it, and all these eccentric experiments one probably would think when they were first planned, leading to this new field of reprogramming essentially. And anything we do and try to manipulate it with our stem cells today is based on his initial experiments and thinking. So that's quite inspirational to be in the within the same building with him. And it was very sad to see him pass away, and hopefully we're organizing something to honor him in the very near future. So that's going to be a nice reunion to celebrate his science and life, hopefully. I did, when he was shutting down the lab, I did steal a little gel comb with his initials on that is in my office. Don't tell anyone. I guess maybe I shouldn't have told this here. So I'm rubbing it for good luck sometimes.
That's terrific. It's a great fit. You're a great fit, and you really, I mean, it's inspiring just even to hear you talk about him, because what people don't realize, or they do, I think that's the great thing about him as just a role model for all of us scientists, is that he really he did it for it. He was so committed from beginning to end. he was unrecognized for many years as, you know, the man, the Nobel laureate, but he was recognized amongst his peers as an inspiration throughout. And his impact on the field before and after the Nobel Prize, the you know, global recognition was, I would say, equivalent. You know, he never stopped, and he still continues to inspire all of us. So a little mini homage, but really speaking to your fit, you know, that was what he did too. You're a great person to carry on the legacy, because he just looked at these systems, and you know, there's a lot of engineering going on right now, but you're still asking these basic questions. You still got that curiosity about how nature made it, and hacks aside. I mean, all the hacks are cool. I love that modern RNA thing, but it's really great talking to you about, you could argue, his science, which he shared with us all. All right, I'll stop about Dr. Gurdon. Rest in peace. We got a couple last questions for you, just to get a little closer, although you really brought us in there. Thank you. First, if you weren't a scientist, which, God, I can't imagine. You probably can't either. What do you think you'd be doing with yourself?
Yeah, I mean, it's a tough one. I think probably I was more strict about this younger, that I can't think of doing anything else. Now I sometimes think like I would have loved to be a farmer. And I think that's becoming a retirement reality as we go forward. But I'm so used to breeding animals now. I need to breed something, I feel. Yeah, no, jokes aside, I mean, I do truly think this was my calling. Maybe something artistic, I would have become a bit more, maybe something like architecture or something. But I did marry an architect in the end, so that satisfies that itch. But yeah, so it's a tough one for me, because I really don't envision doing something else.
Well, you're doing all those things, I guess. A good synthesis of the garden and the breeding, and, you know, you got an architect in there somewhere. Finally, what's the best piece of advice that you've ever been given, professionally or not?
Yeah, so my PhD mentor used to always say, you know, when I obsess over the details, or, you know, in the lab, like, just sometimes we overthink. That's, I think, our occupational hazard, right? He would be like, just do it. Just do it. You know, sometimes, you know, I think this is very important, and just in our science as well, but also in our lives as well. Like, we need to be proactive, and also sometimes take risks. And sometimes you know, be responsible of the consequences, but also it's just not being able to take action is what prevents progress in our science and also in our life. You know, taking that risk, like Gurdon. I signed my Gurdon offer without seeing the place ever. It was COVID. My interview was online. You know, they sent me a video for the lab space. Like, it felt right. You know, it was a risk to be taken, but, you know, just sometimes we just need to go for it. And I find sometimes with my students, and maybe like within the younger generation, they're so scared to make mistakes. It's okay. We all learn by making mistakes. You need to do your due diligence and homework. You need to be prepared, but we just sometimes need to do that experiment. And if it fails, that teaches us something. Or, you know, if we made a wrong decision, we just move on. So just do it with more colorful language sometimes, depending on how angry.
Yeah, it
Was
reminds
also a
me.
great mentor. I need to acknowledge him alongside Alex Dimitris Plakantonakis at NYU.
That's terrific advice. It reminds me of something my mother said to me when I was very young, which I think about all the time. She's still with us. It's never as great as you hope, nor is it as bad as you fear. And that really, every time I do an experiment, I realize that it's not going to be the result I'm exactly hoping for, but it's not going to be nothing, and we're gonna move forward and do something else. So, a bit of a parallel to that, which I think is something that everyone could learn for in science and in life. Sumru, thank you so much again for joining us. This has been a really enchanting conversation for us. I can speak for Arun, I'm sure, and our listeners. We'll have you back again soon.
Thank you. Thanks, Dylan and Arun. This was really fun to be here. Yeah, I was telling you earlier, like listening to you guys when sectioning as a postdoc or walking to lab. So it's fun to be here in a different capacity. So thanks for having me.
All right, that brings us to the end of this episode. What a delightful chat. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCELLpodcast.com, or via email at info@stemcellpodcast.com with feedback or to suggest guests. We were in the neural space, getting insane in the membrane today with Sumru. Not insane, actually, totally sane, sensible, and deep. And we thank you guys for listening. We'll be back in your brains in a couple weeks.